Hey everybody, we are moving on in Unit 4. We're going to start Chapter 17 from gene to protein. And here we're looking at what we call the central dogma of biology. And so this idea that we start with DNA is going to be transcribed into RNA, and then RNA is translated into a polypeptide. And then that polypeptide is a protein, or it could join with other polypeptides to form a larger protein. And so what we're looking at here is just a schematic of sort of where all of this happens. So here we have a very standard eukaryotic cell, very plain, not showing you much detail, but then it does show you this nucleus with the nuclear envelope. Notice that the pores are shown. That is really important because once your DNA is transcribed into pre-mRNA and then it's processed into mRNA, that mRNA can leave through a nuclear pore. And then it's going to be out in the cytoplasm where translation occurs. And with translation, your mRNA is going to be processed or read by a ribosome, and then that ribosome is going to create a polypeptide, which is going to be like a string of amino acids in a row. That the order of the amino acids, that's what's being sort of dictated by that mRNA. It contains the instructions for how to put the amino acids together, and then this polypeptide could be a protein on its own, or it could join with other polypeptides to form a larger protein or a more complex protein. So the DNA that we've been talking about, that is your genetic code or your genotype. RNA is then the message or instructions for the ribosomes to create your polypeptide, which is the product for the phenotype. So your phenotype, or what your physical characteristics, what you look like, is going to be determined by how your proteins are put together, and the instructions from that come from the RNA, and then the genetic code or the DNA is what contains sort of the blueprint for all of your proteins. And then individual genes are going to be read and transcribed into RNA, and then that RNA will be read and translated into a polypeptide. So your genetic code is a sequence of DNA bases that describe which amino acid to place in what order in a polypeptide. And then that genetic code gives the primary protein structure. So when we talk about this genetic code, what is it that we're really talking about? What is the genetic code? So if you were going to say that the genetic code is one base at a time for your DNA bases, so let's say an A, you're looking at an A or an adenine, what you're saying is that one base would then correspond to one amino acid. There's a problem with that though. We only have our 4 DNA bases that we've been talking about, our A's, T's, C's, and G's. And so if we were using just our DNA bases, we would only have 4 amino acids. And that is not enough for our 20 amino acids that we actually know that we have. So if you were to use 2 bases, what that means is that you were reading 2 bases at a time to determine what amino acid to put where. So you could have a combination of A and T, or T and A, C and A, G and C, G and A, any of those. What that would mean is that there were 16 amino acids, but that is still not enough possible combinations. And so when we talk about the genetic code, what we mean is that we are looking at 3 DNA bases at a time to code for 1 amino acid. All right, so an example would be CAT. That is 3 bases read together to code for 1 amino acid. AGC, TTT. Okay, and so when you do that, you end up with 4 to the 3rd combinations, or 64 possible combinations, and that is more than enough for the 20 amino acids that we have. So let's kind of take a look at this and see if we can make a little bit more sense of it. Up here we have a double-stranded DNA molecule, all right, and you can see this gene 1 area is in dark blue. And we are going to look at gene 1 right here first, just this small little segment of it. And so here is your template strand of DNA. So you've got ACC, AAA, CCG, AGT. Okay, that is going to be transcribed into mRNA. Okay, now with mRNA, we don't have thymine anymore, we have uracil. And so we are not going to put a T here, we're going to put a U in place of that thymine. So because we have A here, we have U here, CC would make GG. And then we've got 3 A's in a row, so we're going to have 3 U's in a row. CCG would make GGC, and then AGT would correspond with UCA. So this is now the mRNA segment that we've created, that we've transcribed from the DNA template strand. And now that gets translated into a protein or a polypeptide chain. And so we said that we use 3 bases at a time, so when we look at our mRNA, we are going to have these 3 bases read together, and we call that a codon. So UGG is a codon, and it corresponds to a particular amino acid. We'll get into how you figure out which amino acids go with which codons later on. Right now, what I want you to focus on is that every group of 3 bases in this mRNA transcript is a codon, and each codon corresponds to a particular amino acid. Okay? So the genetic code is based on triplets of bases, and it has some redundancy. So some amino acids have more than one code, and again, we will take a look at a chart that really helps you guys kind of break this down in a minute here. And the proof for this is that we were able to make artificial RNA and then see what amino acids were used in protein synthesis. So this was an experiment in the early 1960s. It is in your book. You guys will have to read on that. But that is the proof for this use of triplets. So a codon is a 3 nucleotide long word in the genetic code. There are 64 possible codons because we have 4 bases, and when you group those 4 bases into 3s, you have 64 possibilities. So really quick, we want to look at what are the differences between DNA and RNA. So we've got a sugar in both. So DNA is deoxyribonucleic acid, and so the sugar is deoxyribose. In RNA, your sugar is ribose, because RNA is ribonucleic acid. So the sugar is a little bit different. Still a 5-carbon sugar. Again, your bases in DNA are the A, T, G, and C that we've been talking about all week. But in RNA, and I already mentioned this, you don't have thymine. So instead you have a uracil in place of it. So anywhere where you would be tempted to put a thymine, you're actually going to put a uracil. DNA has 2 backbones, whereas RNA is single-stranded. It's only got 1. DNA is these really long, very large molecules. RNA is going to be smaller, all right? mRNA is going to be like little segments of those long DNA molecules. You're going to copy 1 gene at a time, so RNA is going to be much smaller than DNA. And then the use, DNA is our genetic code, so it contains all the instructions for all the proteins that our bodies need. Or if you're looking at a cell, it contains all the instructions for all the proteins that that individual cellular organism would need. And then RNA has varied uses because there's different types of RNA. And so we'll get into that in this chapter as well. And this is the sort of instructions that I promised you. So when you are looking at a strand of codons in mRNA and you're trying to figure out what amino acids you need to put where, this is the chart that you are going to use. So if you take a look at this side, it says first mRNA base, the top says second mRNA base, and then the right side says third mRNA base. And so if you were looking at the codon, let's start with AUG. Okay, we're starting with that because that's your start codon, or it codes for methionine. The first base I said was A. So along the side here, you're going to look for A. So A is right here. U was the second base. So we know that we're in this row right here. Now we need to figure out what column we're in. And so U is our second base. And so we're in that column right here. And so AUU, not right. AUC, not right. AUA, nope. AUG, that is what we are looking for. All right, and so you can see it is down here. This is your methionine or your start codon, okay? And so what you're going to do when you get a strand of mRNA codons is you will look each codon up. You'll find the letter, the first mRNA base on the left, the second up top, the third along the right, and that will help you identify your codon and what amino acid that codon is calling for. So the start codon, which we already talked about, is going to be AUG. There are 3 stop codons, and that stop codon, when that is read, it means, okay, stop. Well, you're done making this polypeptide. And so you have UAA, UAG, and then UGA, and they are highlighted right here in that orangey color. And then you've got 60 codons for the other 19 amino acids. So if you look, we're going to flip back. So isoleucine is right here, and you can see that there are 3 codons that all code for the same amino acid. Leucine is right here, and you've got 4 codons here that code for leucine, and then you have another 2 up here. So you can see that some of these are going to have multiple codons for those amino acids. So for testing purposes, you guys need to be able to read a DNA or RNA message and then be able to give that amino acid sequence. You will always be provided with that genetic code table. You will never be asked to memorize those amino acids and what the codons are for each one. All you need to be able to do is take a DNA segment, be able to transcribe that DNA segment into RNA, and then be able to translate that RNA into an amino acid sequence. So real quick, I do want to touch on code redundancy. That third base in the codon shows a wobble. And what that means is that the first 2 bases are going to be the most important in reading the code and giving the correct amino acid. The third base often doesn't matter as much. And so the example with that is really clear here with like leucine, for example. So CU for all of these is going to code for leucine, and so if there was a mistake in that third base, it wouldn't matter because all of these CUs still code for leucine, and so that third base, there's essentially a little bit of an allowance for error based on this redundancy. How has this code evolved? The genetic code is nearly universal. And so an example would be CCG is proline for all life on Earth. And so this means that the code must have evolved really early and that life on Earth must share a common ancestor where this genetic code started and was then passed on to every living thing. So when we read through DNA and RNA, the reading of the code is every 3 bases. We call that our reading frame. And so just in a regular sentence, the red cat ate the rat would be a sentence based on 3-letter words. If you have a frame shift, what that does is it causes improper groupings of the bases. So if we were to remove the E right here from this sentence, the red cat ate the rat turns into essentially nonsense. They're at a tat her at. Okay, that doesn't make any sense. The same thing happens with DNA and RNA. If something happens where a base is removed, it changes the reading frame. It's called a frame shift, and the words no longer make sense. Okay, and so your groups of 3 will be thrown off, and so that can cause some major errors in how proteins are assembled. So we're going to stop here for today. Your lecture question is, in your own words, I want you guys to explain, oh, I'm missing a letter or a word there. Explain why DNA is read 3 bases at a time and how this relates to protein synthesis. All right, so please respond on the website and explain why DNA is read 3 bases at a time and how this relates to protein synthesis. Thanks, guys.